Epistasis. Epistasis is basically a gene interaction in which one gene can affect the expression of another gene. In epistasis, basically two genes are participating. So the expression of one gene or gene pair masks or modifies the expression of another gene or gene pair. So one gene influences the expression of another gene in epistasis. So we have two genes. One is known as epistatic gene, the gene which expresses itself phenotypically. Or you can say the gene which controls the expression of another gene is the epistatic gene, while the second gene is the hypostatic gene which is altered or suppressed by epistatic gene. The gene whose expression is masked by epistatic gene is known as hypostatic gene. So basically, epistatic gene is act as dominantly while hypostatic gene act as recessive. So what is the difference between epistasis and dominance? Epistasis basically is interallelic interaction, means it involves the alleles that affect the alleles that of another gene. While in case of dominance, it is intraallelic interaction, means one allele affect the other allele or hides the expression of other allele that belong to same gene. So in case of epistasis, one gene hides the effect of other at different gene pair. One gene affects the other gene in case of epistasis, while in case of dominance, one allele hides the effect of other allele at the same gene pair. So in case of dominance, the two alleles belong to same gene pair, and in these two alleles, one acts as a dominant and the other allele acts as recessive. The dominant allele expresses itself phenotypically. Recessive epistasis is the first type of epistasis. It is defined as the homozygous presence of recessive alleles at one locus masks the expression of alleles at another locus. So we have two genes that are present at two locus, and if one gene is present with recessive alleles in homozygous condition, then it acts as epistatic gene and it affects the expression of second gene. Its example is the Bombay phenotype, in which the homozygous presence of mutant form of FUT1 gene masks the expression of A and B alleles. As you know, we have I gene that basically controls or blood group system, or you can say ABO blood group system. So I gene has three alleles, A allele, B allele and O allele. So if IA allele is present, it means blood group should be A. If IB allele is present, then blood group should be B. And if both alleles are present, it means blood group should be AB. But in case of Bombay phenotype, although the person may contain the A allele or the B allele, but its blood group is O. So in that case, I gene is basically influenced by H gene. So we have another gene that is named as H gene. H gene basically involved in the formation of H substance. So if H substance is formed, then A allele will convert the H substance into A antigen and B allele converts the H substance into B antigen. So after the formation of antigen, blood group will express itself phenotypically. But in case of Bombay phenotype, when H gene is present in mutant form and its allele FUT1 allele is present in recessive mutant form or homozygous condition, in that case, H substance is not formed. That's why A or B allele cannot express themselves phenotypically. So that is a case, you can say the genotype of this person should be HH with IA or IB allele. So, although A or B alleles are present in that individual, but due to the incomplete formation of H substance, A or B allele cannot express themselves phenotypically. So, the blood group should be O. So, that is a recessive epistasis in which one gene influences the other gene. Another example is coat color in mice. In mice, coat colors are agouti, black, and albino. Two genes are participating in the expression of coat color, and these genes are named as A gene and B gene. A gene is responsible for agouti color when its dominant allele is present, while A gene is responsible for black color if its recessive allele is present. It means if recessive allele is present in homozygous condition, then the coat color should be black. But there is another gene that is named as B gene, and B gene is basically affecting the expression of A gene or controlling the expression of A gene. So here B gene is the epistatic gene while A gene is the hypostatic gene. So how B gene influences the A gene? Basically, B gene is participating in the formation of pigments. So if dominant allele of B is present, it allows pigmentation to occur, while recessive B allele causes the loss of pigmentation. So here are the here is the picture that is showing a cross between the parents. We take true breed parents, that is a agouti, and that is crossed with a albino. So F1 generation is agouti, that is indicating agouti as a dominant phenotype. And in the F2 generation, we have four conditions, four cases, and according to Mendel, the ratio should be 9:3:1 in F2 generation. So here, the first case is showing A capital dash, B capital dash. So here dash is indicating that either the capital A allele or the small a allele may be present here. Similarly, along with the capital B, the second allele may be capital B or the small b, but the phenotype remains unaffected. It means A capital is the dominant allele, so its partner may be a dominant copy or maybe a recessive copy, but the phenotype remains the same. Similarly, along with the dominant B, the other copy may be a dominant B or a recessive B, but the phenotype remains the same. So that's why here dash is present. So 9 cases are there with A capital dash and B capital dash, and its phenotype is agouti. Then 3 cases are there with A capital dash, small b, small b. Here B is present in homozygous recessive condition, while A is present dominantly. So A allele, dominant allele, should be responsible for agouti phenotype, but as B gene is influencing the expression of A gene and B gene is present in homozygous recessive condition, it means no pigmentation occurs, and that is responsible for albino phenotype. The other 3 cases are with small a, and capital B dash. Here capital B dash. So as small a is present along with the capital B. So capital B, dominant B allele allows the pigmentation, and small a allele is responsible for black color. So the phenotype should be black. And the last genotype is in which small a, small b is present. It means that is a homozygous recessive condition for both genes. So here small a is present. It means that should be responsible for black phenotype. But due to the influence of B gene that is present in homozygous recessive condition, A gene cannot express itself. That's why the phenotype is albino. So the ratio for recessive epistasis is 9 offsprings are with agouti, coat color, 3 with black, and 4 with albino. So albino phenotype is indicating that homozygous presence of recessive allele at one locus hides the expression of alleles at another locus. So if B gene is present in homozygous recessive condition, it will affect the expression of A gene, or it will hide the expression of A gene, or in that case, A gene cannot form any color, any coat color. That's why the coat color is albino. So that is the recessive epistasis, and I will discuss other types of epistasis in my next lecture.